It looked like any other perfect day in the South Pacific. All the shallow water creatures, corals and fish, carried on as if nothing extraordinary was about to happen. Then the seemingly innocent rays of the sun struck without warning. UV light zapped their DNA, and the damage was done in an instant. Fortunately, they didn't feel a thing. But for some, their DNA, their template for constructing life, would never be the same. As our undersea sleuths begin solving the mystery of why UV is dangerous to DNA, their counterparts back on land are discovering how DNA itself can help solve some other dark mysteries.
What do we have here? Any clues yet?
Nothing yet, Oxy. Oh, hey.
I want answers, and I want them now.
All right, boss. All right.
Hey, Ox, I think you should see this.
What do you have for me, Dio?
I think I got something here. It's big. It's pretty big. This is gonna have all the answers we need right here. Find out who he or she is.
Good job, Dio. Run a DNA test. Take it to the DNA lab.
Yes, sir. Hey, Ribo. What do you think you can tell us about this?
Well, let's just see what this baby tells us. Oh. Woo-wee, sweet, huh? We're comparing this DNA with our database right now. Okay, boss. DNA results are in. Looks like a him, all right. Male, medium build, brown eyes. Looks like he likes cats, pina coladas, getting caught in the rain.
That's it, guys. The DNA tells the whole story. We're dealing with one big sicko here.
Nuke.
Hey, Nuke.
Hey guys, how's it going? Look, how many times have we seen this on TV? The classic DNA comparison that nails the bad guy.
That's right. We see it and we simply accept it as fact, but not today. Today we're going to go inside the real facts and science of DNA.
Though these might look like barcodes, it's actually DNA, or deoxyribonucleic acid. It's found in all organisms. It is often called the blueprint, or template that defines who and what we are. But before we explore how it works, let's examine what it is.
The letters of the DNA molecule essentially are like a language of biology. And it's composed of 4 different letters, or base pairs. A and T and G and C are the shorthand of what we call those letters. And so the structure of the DNA is what's known as a double helix, which it's double because there are always 2 of these letters that are paired together. And it's a helix in the sense that it twists, like a ladder that's twisted. So we have the different letters here, the different base pairs, are essentially these planes or the rungs of the ladder. And then the pink and yellow colors here on the outside represent what's called the phosphodiester backbone. So there's a common molecule that makes the outer backbone, a rung of this ladder. And then the real information in this language is, again, the bases, these molecules that are in the center, the rungs of the ladder. This is a language of biology because the cell has a mechanism of essentially reading this language and extracting the information that the order of these letters contains. What makes one person a little different from another person is the differences in their DNA.
Now that we know what DNA is and what the molecule looks like, let's talk to a real-life crime lab expert and see what steps are involved in analyzing DNA.
99% of our DNA is the same from person to person.
So forensically, we want to know what's different from person to person. So that's only about 1% of our genome. And an even smaller portion that we're looking at is something we call short tandem repeats. We have 16 locations that we know of, and what we do is go and copy these STRs and see how many times these fragments are repeated at each of those locations. So it's really the compilation of the location and how many times the sequence is repeated, and that's how we determine a DNA profile. So the analysts at their bench will identify a stain. What we want to do is extract the DNA from it, which is just taking it out of the cell nucleus and putting it into solution. The next step is quantification, and we do that on here. It does kind of a mini amplification to see how much DNA is in there. We come back in here with those quant results. And then this machine takes different amounts of the extracted DNA based on the quant and adds it to an amplification. Then we bring the plate into here. This heat block here heats up through a series of temperatures that allow the DNA to split apart, and primers then bind and copy the DNA that's there. Then we need to separate them by size, and we do that on our capillary electrophoresis instrument. Within each of these capillaries, here that are about the size of a human hair, there's gel within there, and the DNA fragments get separated in that very small area. As the DNA fragments pass through, it captures that image.
So, basically, we learned that there are lots of ways that scientists look at DNA from a crime scene. And we wanted you to know that it's not always as easy to solve a crime as it may seem in the movies. So, until next time, we encourage you to never stop exploring your world.
I knew it. The DNA doesn't lie. Those old cars are all jalopy. Those old cars are all humbug. Those old cars are all jalopy. We're dealing with one sicko here.